joining us today. Today I'm presenting on a topic that utilizes a reagent that we sell. So I just want to give you a heads up on that. The AOPI is something normally used on Celometer, but we've had some customers ask, well, can I do this on Celigo? So this is where I want to carry our training today. Is something that may have not been advertised before, but it's something you can do. It's a quick and easy stain. So in the training session today, I'm going to give you a little introduction and background on who would be using this, why are they using this type of application. And then I'm going to show you how to prepare a plate to help you understand the dynamic range of this cell counting place bait method. And then we'll show you how to image and analyze the plate on Celigo, very simple and easy, how to calculate the data and evaluate it for the suspension cell type sampling. And then also I want to show you how to actually lay out a plate sample layout of unknown samples. So you'll see when I talk about the introduction of what I'm talking about for the plate sample layout. All right, so the introduction and the background. So who's doing this kind of assay? So basically, we're wanting to monitor the viability and the sample concentration of suspension cells, but it's not just one sample cell. You want to have maybe 50 samples that you have to measure viability in. So if you're actually counting cells on a celometer or a hemocytometer, those are like one-off samples. So what if you have 50 cell lines? So basically people are doing cell line development, might be monitoring or growing their cells out in shaker flasks or roller bottles, and they need to monitor their viability and cell concentration to see when they need to passage and feed. So this type of application is useful because they have tons of cell lines that they need to monitor. Or in the immuno-oncology world, you're isolating splenocytes from mice or getting patient sample and you need to count your PBMCs, and there's just too many to do in a one-off sample-based method, which would be your hemocytometer with a microscope, or in the automated one sample method, which would be like your celometer or ViCell. These type of applications just become too labor and cumbersome if you're doing one sample at a time. But if you're looking to do something new at a higher throughput level, I want to show you how you can use the Celigo with a plate-based method using the solution provided by Nexlum. It's called AOPI, which is technically acrid orange, which shows up green on your screen or your image, and then propydium iodide, which labels dead cells. So this science about the acrid orange PI is really unique, and you'll see how they work together to provide you a viability stain on the Celigo. So if you're having a lot of samples and they have a dynamic range of something that's very low, like 500,000 cells per mil, all the way up to a very high concentration of cells, such as 8 or 10 million cells per mil, or even more, how do you actually put that in a plate-based method? So this is a training session today to kind of teach you how to apply your suspension samples onto a plate-based method and understand what sample volume you should put in there. And if you have a high range or wide range, what is this dilution factor that you should put in there? So today I'm going to show you some known sample concentrations onto a plate-based method so you can really see the dynamic range of your cell counting. And that will help us or you understand what sample dilution factor you'll need to apply for your suspension cells of unknown concentration onto a plate-based method. All right, so let's get started. So how do you prepare a known sample preparation? Well, first I'm just going to grab a flask from the incubator of known, or today's example, we're going to use the Jurkat cells. And I'm just going to grab a healthy flask, and when I put them on the hemocytometer or the celometer, I'm going to get the cell concentration. Then I'm going to spin down all of the cells in here into a pellet, and then I'm going to resuspend them to a concentration of 8 million cells per mil. So this is my highest concentration that I'm going to detect on the plate-based method today. So then I want to do a dilution series. So I'm going to prepare 5 tubes at the following concentrations. Starting at tube 5, you see it's 8 million cells per mil. But then you're going to have tubes 4 through 1, each being serially diluted twofold. So you got 8, 4, 2, 1, and 500,000 cells per mil. You don't need much. You only need about 200 microliters for each sample. Next, what I want to do, so these would be technically your unknown samples, but these are known concentration samples. So the next we want to dilute out the AOPI, and it's a 20-fold dilution from the stock. And I typically like to dilute it out in a buffer that actually makes the cells happy, but I don't want to use media. So it's going to be, I used HBSS with HEPIs. You can use something else. Maybe your PBMCs are sensitive and you need something that's going to be making them happy, and PBS might not be good enough. So this is going to be the solution you're going to put into a flat bottom plate. So today we're going to use the Greiner 655090 plate. It's a flat bottom, clear bottom black wall plate. And we're going to plate 190 microliters into rows B and C, and then 100 microliters into rows D, E, F, and G. So again, if you're not following me very well, on the next slide, I'll show you the plate layouts for all of these steps next. So then we're going to pipette 10 microliters of the sample set that we have created in those tubes 1 through 5 into rows B and C of the plate. And so for sample 1, you're going to have 4 wells. So B2 and B3, C2 and C3 are all going to be sample 1, and you're just putting 10 microliters into each of those wells. And continue that series to the right all the way out to sample 5. The next, you're going to serially dilute the row C. You're going to take 100 microliters from row C and put it into row D, which contains 100 microliters. So this is serial diluting twofold. And then you're going to continue that serial dilution all the way out to row G. Then you're going to remove 100 microliters from that row G. And so basically what's left is 100 microliters in that well. So visualization, here's some plate maps to describe what I just talked about. So you're pipetting 100, or the AOPI diluted solution into this 96 well plate, and the top rows B and C have 190 microliters. And then the rows D through G only get 100 microliters. So then you can see where the samples that you've created in the small tubes, that 200 microliter volume in those tubes, you're going to put 10 microliters for each sample into the following wells according to this plate map. So you can see sample 1 goes into B2 and 3 and C2 and 3, and then samples 2 are in this group, sample 3 is in this group, 4 is in this group, and 5 is in this group. And it's just 10 microliters per well. And this is what you would imagine like your unknown samples would be plated at. Next, what you're going to do is pipette 100 microliters out of C and put it into row D. And then take D, 100 microliters out, and put it in E, and so forth, all the way out to G. And so G will have 200 microliters, and so you need to remove 100 microliters from G and then discard it. All right. So this plate is now done, and you can see on this last plate map I have is your dilution factor for what we just did, serial diluting the sample set. So row B is a neat sample, which has 10 microliters of our sample suspension solutions. And then we just did a twofold dilution from there. All right. So now we've talked about how to prepare a plate. Now I want to show you how to image and analyze on Celigo. This is pretty straightforward. It's pretty easy assay. So you're going to grab your plate profile, which is the 96 well plate, and we're going to use the Greiner, which is, this list is alphabetical. We're going to look for Greiner 655090. And then we're going to give our plate an ID name. So we'll just type in, I already have one here, it's Viability High Throughput AOPI, and then load the plate. Then we move over to the Scan tab, and we're going to select an application. And the application is going to be in the Viability section called Live plus Dead. It's a 2-channel application. Let me slow this down. So then we're going to select the wells that we want to image by clicking the selection button. And then we're going to click navigation button and navigate the stage over to a focus well. So we'll use C3. Okay, so now we'll set up the live channel parameters to acquire the image in the green illumination. So here we have the live, and then there's the dead, but we're going to set up the live one first. So just change the illumination to green. And then click snaps so we can see what we have displayed in that. Okay, so the cells are there. They're a little out of focus, so you're just going to want to click Focus Setup and then Register Auto. The system will move up and down about 300 microns and try and get the cells in the best focus, and then it will apply the hardware registration focus mechanism. If your cells are way out of focus, you may need to manually move your cells into a little bit better focus plane so that this registration will work. Remember, it's like 300 microns, so that looks really good. So our cells are there, and it's only 10,000 exposure, which is the default setting. And just looking at the pixel intensity of our AO stained cells, we've got about 130 pixel intensity, which is ideal. So if you're in that range where you have your object at around the middle of the camera range, like 130, then the default analysis settings will work really well. And you won't have to adjust too much on the Analyze page. So again, remember the object pixel intensity at 130 is in the middle of the camera range, and that's perfect. Next, we'll switch over to the dead channel for our PI stained cells. And we'll need to change the illumination to red. And then let's click snap and just see what we have on the display area. So you got a few cells, not too many dead ones. They're there and they're a little out of focus, but 10,000 exposure might be good. Let's get them in focus before we change the exposure. So clicking find focus is literally like the computer is going to go and find the right focus, just like we would manually with our own eyes. Well, this is allowing the software to go do it, but it's not going to remember it. So how do we tell the system we want this focus? You can see the current focus offset value is at -33. If we click set offset, that value populates up into this text box area. So now this is telling the system that the red channel should be imaged 33 microns below the green focus channel. That's just where the wavelength of the red versus green actually is in focus. So now we're all set for the red and green channels, and we can click Start Scan. Now we move over to the Analyze tab, and the system will begin to collect the images for the wells that we selected. Now on the analysis parameters, I like to just right off the top, there's a couple of things I know that I'm going to want to check. For both the live and the dead parameters, I want to check separate touching objects and bring up the minimum area to 20. These are round suspension cells, so I know that I'm not going to be looking for, if I have a clump of cells, I want to break it up, so separate touching objects will help do that. And then the minimum area, so is 20. I know the cell object's probably around 30, so we'll set it at 20 as a starting point. So we'll do that for both the live and the dead channel. Those are pretty much the only two that I know for sure I want to set right off the bat. But let's take a look at an image that's been saved on the hard drive and start to evaluate, are the segmentation parameters correct? So here you can see our live cells are in the green image. These are the AO stained cells. And then the dead cells are stained with PI. So a little bit about the chemistry on this stain. AO enters actually all of the cells. It'll stain all of them. But as soon as you add PI to the well, the PI enters compromised leaky membrane or dead cells, and then it quenches the AO stain. So that's why you don't see any AO staining inside the PI cells. So you really get a separation of live from dead cells because the PI will quench the AO. So there is a little bit of bleed through from the AO into the red channel, but it's not nearly enough. You can clearly see the PI dead cells from the bleed through from the AO. So I wouldn't worry about it. If the threshold settings are correct, you're going to be just fine. So the graphic overlay with the default settings are picking up the cells just fine for both the live and the dead. And that's because we picked the illumination that gives us the object pixel intensity in the middle of the camera range. Now we have a few cells over here that are scored as dead, but they're very faint. This might be cellular debris that's been hanging out in the flask. So if you want to get rid of this because you don't truly want to call it as a dead cell, you can actually increase the intensity threshold. The default is at 4, so if we move it up 1, we can start to see things disappear that are just faintly red. Now, if you don't forget to check this little analyze or auto-analyze checkbox. This allows the system to automatically update the parameter on the screen for the values that you're changing on the left-hand column side. So I'm just clicking the up arrow and the graphic overlay will apply it automatically and refresh the display of the graphic overlay. So 6 seems to be doing a good job, still picking up the other bright PI stained cells that looked more like intact cells that have leaky membranes. Okay, so one more thing I'd like to do is go over to the edge of the well, because right now we're looking at the center of the well. I want to click on the edge of the well using the little plate map on the lower right-hand corner, and then drag the image over to see the edge of the well and how is the graphic overlay scoring over here. So you can see the edge of the well is picking up some objects, and it's usually because this is a change in transition from the very dark black wall into maybe some background staining from the AO. So how can we get rid of these long purple outlines on the edge of the well? We can use the aspect ratio. These objects are really long and our cells are round in suspension. So we can just increase this minimum cell aspect ratio until those large, long objects are disappearing. So you can see how they're going away the higher the value is. So just to note, aspect ratio for a cell is length versus width. So if you have a perfectly round circle, the length versus the width is going to be equal to 1. If you have a really long adherent cell, it's going to be less than 1. So by us increasing this minimum aspect ratio, we're getting rid of really long, skinny objects. But you can see how this might not bode well if you were looking at adherent cells. So we have a couple small objects here, not too many. Let's go down a little bit further and see if we've covered the min aspect ratio. All right, we've got a few more long cells here. It's not long cells, it's just the edge of the well is being detected as a cell when it's really not. There's a couple other ways you could get rid of these things, but I try not to grab too many parameters because it might confound other areas of the image. So I just know that the suspension cells are round and long objects we don't need, so we can get rid of it with this one parameter. All right, there we go. That's better. And if we want to get rid of those small little objects up there at the top of the screen, we can increase the minimum area requirement. So we'll bump it up to 30, which is what I would expect a smaller object to be. It has to be that size or bigger. Anything smaller will be eliminated. And again, this will be set up according to your cell type. So PBMCs and splenocytes might be really smaller than this Jurkat cell. So you have to set up the parameters the first time with your cell type. And then once you figure it out, you can save the settings and recall it for your next experiment. So that would be up at the top portion where you click the save button. So this looks pretty good for the live and the dead detection. Now let's click Start Analysis. We'll apply these settings to all of the wells. Okay, here we are on the Results tab, and the images are being still collected, and while that's being displayed, I want to go over a little bit of the measurements that are displayed on the left-hand side. So the data outputs displayed, it starts off with the percentages and then it goes into the counts. So notably, you may be interested in the counts for the live cells, because that's what you're most interested in for diluting out to your cell population. But also I want you to note that there is another one called live corrected. So the third percentage down is live corrected. What does that mean? It's actually incorporating this formula. So you can see that you have live minus live plus dead. What's live plus dead? Well, down here we have a count called live plus dead count, and it's truly double stained cells. And you'll see in this data set today that I don't have a lot of dead double stained cells, so it's not going to affect the number too much. So you'll have live corrected and live. They're not going to vary too much from each other. But why is this important? For example, today I don't have a lot of double stained cells because I think the cells are happy in this PBS, or excuse me, the HBSS sample. But if you have cells that are dying in the process of doing all this, you might have a lot of cells dying in the plate. So don't forget to save your experiment. This will allow you to recall it when you run your next plate of unknown samples. So at the bottom of the screen at the lower left, you just hit the save button and you create a file name for it. And then when you load your next plate, it will remember everything, the application, the illumination, the focus, the analysis settings, everything is remembered. So you can just use that whole experiment over again on the next plate. Things are adjustable if you use those save experiments. Say, for instance, you want to include every well, and we've just done the inner wells. You can make that adjustment. So let's look at one of our wells that have the lower concentration, but it's a neat sample, meaning 10 microliters into the well. So you have a fair amount of cells. They're properly segmented. It's looking really good. It's not too dense. So you can see that the dead cells and the live cells are properly segmented. And you can turn on and off the graphic overlay and see how what it's picking up. And it's doing a good job. And some of the clumpy cells are being segmented properly. So it's doing a good job for the slightly clumpiness that we do have. That's the separate touching objects part of the algorithm. It's doing a good job for that little cluster right there for these lower cell densities. But now if we look at the cell densities that are higher, so let's move over on the plate and look at the higher cell densities. Okay, so our dead cells are looking good. And if we move up over into, so maybe the 2 million cells per mil, this would be 10 microliters into 190 of the AO solution. So you can see here's the dead cell count, and then here's the live AO cells. So it's getting more dense, and I think it's still doing a good job detecting the cells. Maybe a couple of clumpiness that's not being picked up, but I think it's doing a good job overall for this cell density. And then if we go up further to maybe 4 million cells per mil density, let's see what does this look like. All right, so for the most part, I would say 90% is doing a good job. There are a few clumps that we're missing, and it might be just getting on the edge of our threshold of detecting the cells. The clumpiness is not being picked up, so this might be too many cells in a well to properly count. But that's the whole point of doing a serial dilution. We need to figure out for this cell density, what is the proper dilution for our cell densities. So here we just did a serial dilution of that population, and it's doing a better job. There's a few cells that are still clumpy. Let's go one more down. So this would be fourfold diluted from our neat sample. All right, that's looking a lot better, much more similar to our maybe million cells per mil. That's kind of more of what it's looking like, or the 2 million. So I would have confidence this is looking really good for segmentation and counting of the cells in the well, and it's not missing the clumpiness because there isn't much. Okay. And now we're at the edge of the well, and that's typically where the cells can struggle or they end up clustering over at the edge of the well. So let's look at the highest density at the edge of the well and see how does this image look. All right, so here we are at the 8 million cells per mil, and we're missing a lot more cells on the edge of the well wall where they like to congregate. So this might be too concentrated of a sample to detect just by pipetting 10 microliters of the sample straight into the well. So if you have samples at 8 million cells per mil, you may need to do some type of dilution into the plate-based method. Now let's export this data into a CSV file and open it in Excel. And I want to show you how you can look at this quantitatively to help you determine what is the best dilution factor for your sample set of unknown samples. Okay, so we've exported it and saved it on the desktop. Now we've covered imaging and analyzing. The imaging and analysis is pretty easy. It's only 2 channels. If you do the illumination right, the default settings work on the Analyze tab. Now I want to show you how to calculate for the cell concentration, cells per mil, and figure out what is the dilution factor for a large range of unknown samples. So in the CSV file, the data displayed is same as what we saw on the results tab. You have your percentages showing up first, which is your percent live, percent dead, percent live corrected, which is removing the double stained cells. And you can see further down we have the live count. So those are items detected with the AO staining, and then the dead is for the PI staining. And then the live plus dead count is actually objects that were scored for both the PI and the AO. So what we call those are double stained cells or double positives. And just looking at the counts, you can tell there aren't that many. But I know that I've heard some people say, well, I've got a lot of double stained, and I kind of quickly realized that it was probably the buffer that they put it in, which is PBS, and maybe the cells were not happy in that PBS buffer and they needed a little bit more components in there to go through this counting their more sensitive cells. So that's why I switched this to HBSS with HEPIs. Okay, so what I want to show you is how to calculate the live corrected counts and the total counts, so utilizing some of these values. So on this page, it's just the CSV content, but on the next tab over, I've created a calculations tab that's going to grab data from this page. So, okay, in here at the top, I have this live corrected count. And how did I figure this live corrected count? So this is actually using the live counts minus the double positive stained cells. And that's all this formula is. And it's just grabbing the information from the other page. And that is truly our live only cells. And then the dead count is just grabbing the data from the other page. There's no formulas involved. It's just grabbing the data raw from the dead count section. And here is the calculated total. So this one is actually utilizing the live only or live corrected count plus the dead count. So this is giving you a total cell count. Now, depending on your application or your protocols in your lab, you may be only interested in that live only count and you don't really care about the dead counts. Maybe you're just interested in the percentage of viability. So just depends on what value you're most interested in. All right, so let's do some analysis on how did we do with this plating and detecting of the cells in the well with the different concentrations. So here I have actually created a table of the theoretical values that I was expecting versus the actual, and I'm just averaging the duplicates in the plate. So for this grouping here, which was the 500,000 cells per mil, for the top neat sample, I was expecting 5,000 cells, but I actually got 6,000 cells. So that could be, again, to my plating, my maybe not so perfect plating. And so I do want to comment on that. If your pipettes are not calibrated, or if you have bad pipetting technique, this can throw this whole situation off. So I highly recommend that you have calibrated pipettes and you have a good pipetting technique. So here I've just plotted the theoretical versus actual for each of those samples and put them on the graph and created a R-squared value for each of the groupings. And so you can see the lower density with the serial dilution is actually doing a very linear, so this means I was linearly diluting out in my plate just fine. So diluting out in this plate just was fine. And then the next cell population down is 1,000,000 cells per mil, and that's in the orange color. And so the R-squared value for the 1,000,000, the 2,000,000, and the 4,000,000 actually are the highest R-squared values. And you can see where the lowest value, 500,000, and the 8,000,000, it's still good, it's 0.99, but that's where you start to fall apart. And it could be that we're just too low on the low end with the diluted out 32-fold dilution for that low end sample, or on the very high end where the 8,000,000 cells per mil we plated neat is just too many cells in the well to actually accurately count. So this is just to kind of help you understand your sample plating and how you should dilute your samples of unknowns. So this cell concentration is a cells per mil formula. So I'm factoring in the dilution factor, the volume of the sample, and then the actual total cell count. And then I'm multiplying it by a mil, so you have a cells per mil concentration. Okay, so then we just put in 10 microliters. So you would just adjust this formula to whatever volume you end up putting in there. I'm most comfortable pipetting in 10. Some people are better, maybe would prefer 20, or if you wanted to do 5 microliters, you can do that. Just make sure you account for how much volume of your sample you're putting in your well. So if you actually factor in the dilution factor, you would probably get you would get this series where you're supposed to be hitting the same value. So if you look at the average for that grouping and the standard deviation, you can see that the low end, I have an average set here, and the standard deviation is on the is kind of high for the low end, and even the CV shows it. So there may be a sample down there. I think it's the last one with the 32-fold dilution. If we actually eliminated that data point from the group of averages, we can improve our CV because it's so different from the rest of them. So this kind of shows to you that maybe this is too dilute of a sample for accurately counting the cells. So it did improve our CV. So let's go and look at what was the actual count for that well. Maybe these two are probably too low. Yeah, it got below 15, which is better. But I just want to see what was the cell count so we can kind of see what was the range of the cells in the wells. All right, so we're about 200, 300 cells, about 100 cells. So that might just be too few of cells to accurately count what is the proper cell density to back calculate if you have a sample that's at 500,000 cells per mil. Now, how about on the higher end? Let's look at the 8,000,000 cells per mil. And here you're in the highest end where you have a neat sample. And you would expect around 8 million, but no, it's just below that. So maybe the neat sample at 8,000,000 cells per mil is not exactly correct. What was the value in the well itself? So it's only at about 40, 60,000. So we're probably on the top end, not accurately counting the cells when it's just plated neat into the plate, 10 microliters. So you can see where we're kind of topping out or plateauing for our count. So we have this low end that might just be too few of cells to have a count of accuracy. And then here you have on the high end, just a little too few cells, or too many cells because they're clustering and clumping together to accurately count that. But when you dilute it out into the plate, you have a better chance of hitting the correct accurate number. So what would be a good detectable, or what would be a good dilution factor for your samples of unknowns? So I would pick something in the middle. So we have 4 and 8 seem to do well for both. So then I would probably do some kind of dilution factor like that into my unknowns. So if you back calculate that value, it becomes a final dilution factor of 100-fold, because I put 10 microliters into 190, that's 20. And then if you did 5 as a dilution factor in the plate, that became 100. So now let's put a plate layout together, showcasing how you can do that with unknown samples. So that's our goal. We're going to make a dilution factor final into the plate for imaging of a dilution factor of 100. So we're actually going to use 2 plates in this sample prep setup. So first off, we're going to make a 20-fold dilution of the AOPI, and then we're going to prepare 2 plates with the AOPI solution. So in the first plate, it's going to be a U-bottom plate, and we're putting in 90 microliters of this AO solution. And in the second plate, we're going to put 90 microliters into the imaging plate, which is the Greiner 655090 plate. So what we're going to do is initially plate our samples into the U-bottom plate containing 90 microliters. So you're basically pipetting 10 microliters of sample into this pre-dilution U-bottom plate. That's the top plate up here. And then what you're going to do is you're going to mix that, so 10 microliters plus 90, mix that in the U-bottom plate, and then you're going to take 10 microliters from this solution in the U-bottom plate and pipette it into the imaging plate below, but you're going to do that in triplicates. So you can see in row A, 1, 2, and 3, you've got the same sample just plated in triplicates. So that'll give you your average and standard deviation, or if you have a pipetting error, you have the ability to remove. So you're going to do that for your whole sample set, and you've got 39 samples so you can plate in this situation. And then you're going to spin your plate down just real quick to settle the cells at the bottom, and then go ahead and image it on the plate on the Celigo. So at this point, you can recall your saved experiment settings that you saved from our testing of the linear range for the unknown samples. So that's just an example of how you would actually image the plate with unknown samples. All right, so we've covered a lot today, some of the background of what kind of application this is and who would be using it. And when it's a lot of unknown samples that you're culturing in suspension, it's a very helpful, quick way to access the viability and concentration of cells. And then I showed you how to actually prepare a plate to understand the dynamic range with your cell sample set, how to image and analyze the plate on Celigo, and then how to look at the calculations and evaluate the data to figure out what is your best dilution factor for your sample set range. And then I provided an example how you would actually put the plate layout together for your unknown sample set. All right, so that's pretty much it. I covered a lot, so if you have any questions, just let me know and we can answer them for you.
Thanks, Sarah. I think that was a really great presentation. So as Sarah said, we are opening this time up for any questions that our attendees have for us. So I'll give you a minute or so to type those in. And while we're waiting, I just want to let our attendees know that we do encourage you to submit some topics for ideas for some future webinars. So if you do have a certain topic that you'd like to see covered on the Celigo, you can submit that to us at support@nextlim.com, or you can fill it out on the survey after this webinar, and we would like to review your suggestions. Okay, So we do have a couple questions here for Sarah. So the first question says, how long can the cells sit in the stain before they begin to fade?
I don't know how long it takes for them to fade because I was pretty much done. Once I was done imaging, I was done with the plate, so I didn't let it sit around. I didn't see longer staining. And actually, if I imaged it twice, like one time I did a pass just right after coming out of the tissue culture room, I imaged it. And then I imaged it again. It almost seemed like the AO got a little bit brighter, so I felt like I could turn the exposure down. So I would say within 30 minutes of me plating the cells, I probably had it go a little bit brighter, not fader.
Alrighty. We do have another question. So does the AOPI solution have to be made fresh each time? And can the plates be prepared in advance and pulled out when the cell counting is about to happen?
I have only made it fresh. I haven't made a diluted sample and let it sit for 24 hours and then used it. I haven't done that, so that's a good question. I would imagine that the dye solution, if you kept it diluted, would be good as long as it's kept in the dark and maybe cold. But then when you prepare your cells, you're going to want to bring it to room temperature. As far as putting the samples, I'm not quite clear on the second part of the question about, I'm not quite clear about putting the sample set up. Can you maybe rephrase the question, the second half, where you put the samples in and then image it? How long? Can you ask the question again? Uh-oh. I have technical difficulties. Hang on. I lost the call. The phone hung up. If you can. Oh, okay. All right. It looks like Amy's having technical difficulties, so we're going to have to